Good afternoon, everybody, and welcome to the prize lecture this afternoon. It's my great pleasure to introduce Robert Tarran as this year's recipient of the Hodgkin-Huxley-Katz Prize. This prize is named after the 3 Nobel Prize winners for their study of nerve impulses and neurotransmitter release. And they used electrophysiology and studied the role of ion channels involved in these processes. So in keeping with the ion channel theme, Robert Tarran first experienced electrophysiology and patch clamping techniques, I understand, in his undergraduate degree at Leeds, and this piqued his interest in ion channels. He then went on to do a PhD with Mike Gray in Newcastle and began his interest in epithelial transport and the cystic fibrosis transmembrane regulator. He completed his postdoctoral research at the University of North Carolina in Chapel Hill and also spent some time in the University of California at Berkeley, where the group showed in North Carolina that the lack of cystic fibrosis transmembrane regulator mediated ion transport in the lungs resulted in dehydration of the airway surface liquid. And this underpinned the pulmonary pathophysiology of CF disease. Rob on his own then went on to show that the lack of CFTR also caused an environment that was favourable to the growth of anaerobic bacteria in the lungs, and winning the Adolf Window Prize for that work. So Rob's still currently a faculty member in the Department of Cell Biology and Physiology at UNC. He's published extensively on the role of ion channels, including the epithelial sodium channel and the CFTR in the airways and the pathogenesis of CF disease. Over the last 5 years, he's been studying the effects of cigarettes and e-cigarettes on the lung, including on ion transport, and has published some of the first physiological studies on the effects of vaping in human lungs. He was responsible for obtaining $20 million worth of funding for Tobacco Centers of Regulatory Science, and Rob has chaired FDA committees and workshops on new and emerging tobacco products, and chaired and contributed to a committee and contributed to a white paper on e-cigarettes and nicotine for the NIH. But in reference to the work of the Hodgkin, Huxley and Katz, today he's going to be talking on how peptides can modulate ion channel physiology. So over to you, Rob, and welcome.
Okay, thanks. So I'd like to thank the PhysSoc for this award, and thank Debbie for that lovely introduction. So I'm going to be talking about Splunk1, how it can regulate cation channels today. So here are my disclosures. But Debbie suggested I give a couple of slides of kind of where I came from and how I got to where I am now. So this is me as an undergrad. I kind of meant to dug this one out of the vault. So I'm from here and kind of from here. And I like the mountains. So I went to Leeds for my undergraduate and then Newcastle for my PhD, as Debbie said, so that when I wasn't doing electrophys and learning about physiology, I could play in the mountains. And that was part of the reason I went to North Carolina. So then I went to North Carolina in '97. My plan was just to go for a year or two, then come back to the UK. But it turned out that North Carolina is actually a really cool state, so I ended up staying. And there's loads to do there, which kind of aligned with my interests. Plus, it's a great research center as well. And as Debbie said, I've had a long-term interest in ion channels. And I think when I was starting out, I was drawn to the kind of simplicity of them. At the basis, they're holes in the membrane which open and shut and let ions through to exert their physiological processes. And then also, as Debbie said, during my PhD and postdoc, I studied cystic fibrosis a lot. And I'll talk a little bit about CF today, but also about other diseases, and then also focus on the ion channel physiology of Splunk. So cystic fibrosis is caused by defects in the CFTR. This is the molecular structure of it. And these defects caused altered epithelial transport in a number of organs shown here, including the pancreas, the GI tract, and the sinuses. But I focused on the lungs, which now represents one of the biggest problems for CF patients. During my postdoc, I was part of the team that identified the lack of CFTR chloride channels and maybe too much sodium channels caused this dehydration that you see in CF lungs. And then this really actually produced the link between the observed physiology was too much mucus in CF patients and people that had a hard time reconciling that with altered ion channels. And this kind of made the link. So, but before I go into Splunk physiology, I want to back up a little bit and just talk a little bit about the airways and their anatomy physiology. So the airways can be broadly divided into the conducting zone and the respiratory zone. In the conducting zone, you have one trachea or trachea, and then a few bronchi and bronchioles. The numbers of them are here, so 1 down to tens of thousands. And this has the surface area roughly the size of a tea towel. In contrast, the respiratory zone, which they're here up to many millions of alveoli where gas exchange takes place, has a surface area the size of a tennis court. So the lung is quite big if you were to unfold it. And because of this, we breathe in about a cement mixer-sized truck of air every day. And as people have become aware in the last year or two, there's not only a lot of bacteria, so everyone's typically exposed to 60 million bacteria during their breathing throughout the course of a day, and a similar number of viral particles. I think what's really interesting is in the gut, there's 10 to the 9 to 10 to the 12 bacteria per mil of luminal contents, and there's no immune responses, your gut does fine. But in comparison, and this is something I show my students, so this is from a textbook. So it doesn't matter if you're in a laboratory where there's 200 bacteria per cubic meter of air, or in a sewage plant where there's nearly 1 million bacteria per cubic meter of air. By the time this air has been breathed into your lung and reaches the alveoli, it's been cleaned and sterilized, and all these bacteria have been taken out by the lung. And this is happening with every breath, and this is innate defense in the lung. And so I really kind of study how ion channels regulate innate defense in the lung. And this can happen in 2 ways, both of which I'll talk about today, by blood cells. So typically 98% of the white blood cells, which are hanging out in the lung in a non-infected person, are macrophages, which can engulf bacteria. And then also, so the epithelial, which line the lung, have ciliated cells, and they can use CFTR and the sodium channels ENaC to secrete fluid and help clear out mucus by ciliary beating. And this is a cilia beating over the epithelial cells here. So I guess I try to understand how ion channels can regulate innate defense in the lung. Going in a bit more detail, so this is a cartoon from Felix Ratchen showing cross-section through a large airway. So here are the ciliated cells which beat and move mucus out of the airways. And you might be more aware of mucus being kind of cleared from the airways if you have a cold, but actually mucus is being produced and secreted with 24/7, as it were, and you're swallowing it without even realizing it. So Mehmet Kesmer and did some really nice proteomic studies. And so this kind of airway surface liquid and mucus has about 1,000 proteins in it. And this is from Mehmet. So this is showing what he thinks some of the functions are. And so these are just predicted functions, but really for a lot of these proteins, we actually don't know what the function is. So there's a lot to do in understanding why there's 1,000 proteins in the airway surface liquid which lines the lung. However, splunk, which I'll go into a bit more detail in a moment, is actually present in this lung lining fluid. And I think from this situation, it can interact with the sodium channels and also interact with calcium channels. So this is splunk. When we got interested in how splunk could regulate ion channels, splunk had been found, but no one has had really assigned a function to it. So splunk is highly expressed in the lung, and it's secreted into the lung lumen, like I mentioned. So if you look in the literature, it's also known as plunk. My personal favorite, spurred, which is secreted protein from upper respiratory tract, lungs, and then the gene name is BPIFA1. It's been shown over the last few years that splunk is actually a gene modifier both for cystic fibrosis and for asthma. The work I'm going to talk about today is how it can regulate ion channels, and then I'm also going to talk about how we can use this understanding to make new peptide-based ion channel inhibitors. So in collaboration with Matt Redenberg, in chemistry at UNC, he solved the crystal structure, and this is splunk. And Matt joked that because it had different functions, it was like the Swiss Army knife of the airways. And so I'll talk today about how it can regulate the ENaC sodium channel and also the ORI calcium channel. We and others have shown that it has antibacterial functions, which we can assign to distinct domains of the protein. And then Peter D. up in Pittsburgh has data suggesting that when splunk gets degraded, it acts as a neutrophil chemoattractant to basically encourage neutrophils to come into the lung during infection. So I'm going to show a little bit of clinical data, which actually came out after we'd studying it, but I think this sets the context. So this is data from both normal and cystic fibrosis patient sputum. So this is airway secretions, but then also from asthma patients. And then this is the mean data over here. So this is splunk in normal subjects. So splunk levels are actually down quite significantly, both in cystic fibrosis lungs and then also in asthma patients. And then this is allergic patients who don't have asthma as a disease control where splunk levels aren't down. Now data suggest, so this is taking recombinant splunk and putting it into sputum from normal subjects or cystic fibrosis subjects. And you can see that while it's stable actually over 24 hours in normal sputum, it's rapidly degraded with a half-life of 20 minutes in cystic fibrosis sputum. And Megan Webster, who did these studies, actually found that it's neutrophil elastase, which is a protein that's released from neutrophils when they come into the lung, that's responsible for most of this degradation. And I'm not going to show the data, but we and also Hongwei Chu's group from Denver also showed that IL-13, which is a cytokine that's up in asthma. So it's a different mechanism, but will actually drive down Splunk expression. So there's two potential ways that Splunk, which we think is important for the innate immune system, can kind of go away during times of disease, which is probably not a good thing. And this is actually some data from Clemente Britos at Yale, which we collaborated with him. And he followed cystic fibrosis patients that had either higher Splunk levels or lower Splunk levels. And this is the data over 60 days, but he followed them over a year and saw the same thing. And basically, CF patients who had higher Splunk levels actually exacerbate less, so they stay healthier and go into the clinic to be seen less often. And then also Hongwei and his group also showed that in asthma patients, those who have higher Splunk levels actually have better lung function. So I think this clinical data suggests that Splunk is important in the lung and is worth studying. So now I'm going to go into the ENaC part of the story. So the epithelial sodium channel ENaC is a heterotrimer that's expressed in the lungs, gut, kidneys, amongst other organs. So this is ENaC here. You need to have the three subunits, alpha, beta, and gamma, together to form the sodium conducting pore. And it's highly selective for sodium over potassium. So in normal lungs, we think that ENaC helps regulate the amount of fluid in the lung. So you have a little bit of fluid in the lung, which is secreted and then moves up the lung by mucus clearance and by cough clearance. And then, so you don't have too much fluid, excess fluid gets absorbed by ENaC. And increased ENaC activity is thought to be contributory to cystic fibrosis lung disease. So some of the work I did during my postdoc, Scott Randell was able to grow primary lung epithelial cells. These are taken straight from the lung and from the bronchial region and plated down. And then after about a month under air-liquid interface conditions, which is a fancy way of saying there's culture media below the cultures, but nothing on the top, and the cultures make their own secretions. They form about 80% ciliated cells, which are shown here, and then also have these about 20% goblet cells, which secrete the mucins. And then they have basal cells beneath them. And so we figured out that we could put fluorescent markers into this airway surface liquid, which is this kind of like space above the cells. And then we could put them on a confocal and measure it, which is shown here. And so this is an actual image. And then we could label the cells with calcine if we wanted to. And because this layer is only about 7 microns thick, we would put some perfluorocarbon on top, which is immiscible with the airway surface liquid, but is oxygen permeant. So we could then measure this on the confocal microscope. And we could also put electrodes into this thin film and measure the transepithelial voltage at the same time. But unfortunately, because of the cilia and the mucus, we couldn't patch these cells, not when they were intact. So these are some of the studies I did. So here we're adding, we're adding a fluorescent dye just above the cells and measuring the height of the airway surface liquid, which is shown here in microns. And then we're tracking this over 24 hours. And so we added equal amounts of fluid to both normal and CF cultures. And the normal cultures absorb excess fluid and then stop absorbing and maintain ASL height at 7 microns, which is roughly at the height of outstretched cilia. In contrast, the CF cultures kind of don't know when to stop and basically keep absorbing until all the fluid is gone. So when we did the microelectrode studies, what you see is, so normal cultures, they have a moderately high voltage, they elevate it a little bit, and then it declines over time. Whereas in CF cultures, it stays significantly high throughout the whole experiment. And what's interesting is at the start, when there's absorbing, there isn't actually a big difference between normal and CF cultures. But then at 24 hours, when normal cultures are finished absorbing, there's a significant difference. And I'm not showing the data, but we know that the decline in voltage is due to a lack of ENaC activity. So the normal cultures can sense the volume and turn off ENaC. And we were interested in trying to find out how that happened. And my reasoning was ENaC needs to be proteolytically cleaved to conduct sodium. So I thought there was going to be a soluble protease inhibitor, which is accumulating in the airway surface liquid, which was then turning off ENaC in normal but not CF cultures. And so at this point, I hadn't done very much biochemistry and had really focused on electrophysiology and confocal microscopy, but I got help from people at UNC like Jack Stutz, Scott Donaldson, and we collected airway surface liquid from cultures. We pulled it, and since we were looking for a protease inhibitor, we incubated that with trypsin-coated agarose beads at 4 degrees with basically non-trypsin-coated beads as a control. We did SDS page and cut out bands from mass spectrometry, and really the only hit we got was Splunk 1. And that's how we got into studying Splunk and ENaC. And it turns out Splunk isn't a protease inhibitor, but it does regulate ENaC quite well, I think. So then we did some oocyte studies. So we expressed ENaC, cDNA plus minus proteases in oocytes and did 2 electrode voltage clamp. And so this is some of the data. So this is basal ENaC current. And if you co-express a protease, so this is channel activating protease 2, you get an increase in current. And under both conditions, so if you compare this to this, there's about a 50% reduction in ENaC current. And then even in the presence of the protease, again, there's a probably greater than 50% reduction in current. And we saw this also in HEX cells. So there was good evidence that Splunk could inhibit ENaC in heterologous expression systems. We also knocked down Splunk in the primary airway cultures. And these are the cultures, and we had paired cultures, either with or without Splunk. And knocking down Splunk led to CF-like levels of airway surface liquid in these cultures, again, suggesting that, at least in the cultures, Splunk was physiologically relevant. And then we could add back recombinant Splunk and restore this volume. And I'm not showing the data today, but we also did microelectrode studies to confirm that we were affecting the ENaC mediated voltages. And my title mentions allostery, but the way the talk went, I apologize. I took out all the allostery data. And then, so Christine Kim, he was a graduate student in my lab. I'm basically reducing her whole graduate studies into one kind of cartoon slide. And if Christine's watching, I apologize for that. But she published data a couple of years ago in FACIT, and what we think is happening is Splunk combined to the alpha beta gamma ENaC subunit. Splunk binds to the outside, and by using FRET-labeled constructs, we can see changes in FRET on the inside of the cell, which indicates allostery. So Splunk's causing a conformational change in ENaC. We then found there was NED42 dependent ubiquitination of ENaC, which then caused internalization of some of the ENaC subunits. And if Christine expressed a dominant negative NED42, you actually block the Splunk-induced internalization of ENaC. We also found that Splunk sent ENaC to the lysosomes, and if we use chloroquine to block lysosomal acidification, you actually no longer saw the degradation of Splunk. So for a series of papers, I think we could demonstrate that Splunk did inhibit ENaC, but rather than being a traditional pore-blocking ion channel antagonist, it actually binds to the channel, internalizes it, and then sends it for degradation. We then did some studies to try and see which part of Splunk was inhibiting ENaC, and these are studies by a graduate student, Brett Rollins, in the lab. And what he did is using the cRNAs to inject in oocytes, he gradually deleted more and more of the cRNA starting from the C terminus. And he found that you could actually delete up to 85% of Splunk and still get full inhibition of the ENaC current. And then if you went a little bit over that, you lost the effect. And at the time, so we know that the first 20 or so amino acids were an N-terminal signal sequence to get Splunk secreted, that we assumed that this green region was the part that was responsible for Splunk. And historically, because it was from Splunk and we thought it had 18 residues or 18 amino acids, we called it S18 and the name stuck. So if you hear me talk about S18, I'm talking about ENaC's, I'm talking about Splunk's ENaC inhibitory domain. So moving back to the primary airway cultures. So these are normal cultures which have a 7 micron ASL height, and these are CF cultures which have significant dehydration. When we added recombinant Splunk, we saw an increase in ASL height in the normal cultures, but Splunk didn't work at all in the CF cultures. And we tried this several different times, and we saw the same thing. What was interesting was when we made this peptides, which is the corresponding to the N-terminal region of Splunk, the peptide worked both in normal and CF cultures and elevated ASL height. So recombinant Splunk doesn't work in CF, but the peptide just of the ENaC of the N-terminal region did. And this is just what some of the raw data looks like. So this is a CF culture. The red is the airway surface liquid that's been labeled with fluorescent dextran. And you see this kind of classic dehydration. And when we added the S18 peptide kind of apically, you see that the height of this layer is thicker, which is indicating that you've blocked ENaC and you're preventing dehydration. And we thought about this and what could be different from the normal versus CF cultures. And we didn't think it was going to be effect on, we didn't think it was due to just abnormal regulation of ENaC because S18 still worked. And Ray Coakley, who's an adult pulmonologist in UNC, who's a longtime collaborator, he pointed out that there is supposed to be a difference in pH in the airway surface liquid between normal and CF subjects. So he obtained samples from 12 normal and 12 CF patients. And he actually measured the pH, and you saw there was about a half a pH unit difference. So CFTR doesn't just conduct chloride, but it conducts bicarbonate. And the idea is there's less bicarbonate going into the lung lining fluid, so you get a very moderate acidification. He also, I'm not sure how he got them to agree to this, but he also got the subjects to chew chili peppers, which then made their eyes and their nose run. This actually stimulates glandular secretions. And under those conditions, you actually didn't see a difference in pH. And he took from this that the difference in pH was due to the airway surface liquid, not due to the glandular secretions. So we then looked at fluorescent splunk binding to CF cultures. And you can see, and this is basically just the amount of binding. And there's very little binding at pH 6 or pH 6.5. Then once you jump up to pH 7, you get much more binding. And we saw the exact same thing on normal airway cultures. So splunk binding to ENaC didn't care about the presence or absence of CFTR, and it just cared about pH. And in contrast, the S18 peptide, when you looked at binding to that, to CF cultures, it was pH independent. So this fit with our kind of more physiological observations that splunk couldn't inhibit ENaC under kind of open circuit conditions, but the S18 peptide could. So then Matt Redenbaugh and Sompot Bencharat, who are the structural biologists, they thought about this and they knocked out all the histidines in splunk because histidines have a pKa of about 7, and that didn't do anything. And then they thought about it some more, and they identified these salt bridges on splunk's surface. And salt bridge formation can also pH dependent. I'm not showing the data, but it's published, and what they found out was if you mutate out any of these salt bridge residues, you actually block the pH dependency of splunk and make it pH independent. And what was quite neat was they could actually switch the position around of these salt bridges and actually restore the pH effects. So what we think is happening, so this is the splunk which Matt and Sompot solved the structure of. And then they've added on the S18, the ENaC inhibitory domain, which is disordered and doesn't have any structure. And we think that under pH 7 or above conditions, the S18 region is extended and combined to ENaC, shown here. But then in a pH 6.5 or below, we think that the S18 interacts with the cross bridges and is no longer available for binding. So unfortunately, we weren't able to, splunk wasn't amenable to investigation by NMR. So we haven't been able to directly visualize this, but this is the best theory we can come up with. The other thing which I thought was interesting is when Matt tried to make crystals of splunk, you could crystallize it at pH 7, but it wouldn't crystallize at pH 6. And the other thing he tried is he crystallized at pH 7, then infused the crystals with acid, and apparently the crystal just all broke apart, which is indicative of movement in splunk, which again supports this hypothesis. So we also did some in vivo studies, and in sheep, S18-derived peptides actually restored changed airway hydration and increased mucus clearance, which is consistent with the mechanism of ENaC. And then I'm not going to talk about it today, but we were actually able to get FDA approval to put S18-derived peptides into CF patients. And in some patients, it gave a really large increase in lung function, but in other patients, it didn't work so well. So I don't think we fully understand why that was. But broadly speaking, the in vitro data are consistent with what we see in vivo. So I'm going to switch gears now and talk about the ori calcium channel in Splunk1. So the ori from Greek mythology are the basically responsible for the changing of the seasons and are the gatekeepers of Olympus. I think the people that study ori calcium channels think it was really important, so they named it after the ori. And so this is also a plasma membrane ion channel that conducts calcium, not sodium or potassium. And basically, when there's a second messenger cascade, STIM1, which is an endoplasmic reticulum protein, will move to the endoplasmic reticulum plasma membrane junction, activate ori, which then lets calcium in. And ori is really important, for example, in the immune system, where it can drive inflammation and airway remodeling. So the reason we got into ori is another collaborator of mine at UNC, Steve Tilley, was studying the Splunk knockout mice. And the Splunk knockout mice initially were a little bit of a disappointment. So they didn't have a major phenotype. They show some hyperinflammation if challenged, and they also are more susceptible to bacterial infections, but nothing which some people thought was earth-shattering. But then Steve noticed that these mice had spontaneous asthma. So if you put them on a flex event, which is a way of measuring lung function in mice, and gave them methacholine, which is acetylcholine analog, you saw basically greater bronchospasm or airway hyperreactivity in these mice. So suggesting they had basically spontaneous asthma. And we thought about this, and we thought the only way that could be happening is if Splunk, which is shown here as this green dot, was actually inhibiting calcium influx into the airway smooth muscle and causing relaxation. So in airway smooth muscle, basically contraction or relaxation basically tracks with intracellular calcium levels. Now, we thought about this, and I must admit I was skeptical at the time. But then Ton De Woo, who was a postdoc in the lab, got primary human airway smooth muscle and did a dose response and used FURA2 to measure intracellular calcium, and found that Splunk actually caused this dose-dependent inhibition of calcium signaling in these airway smooth muscle cells. And I'm not showing the data today, but she could also show that Splunk blocked contraction both of isolated airway smooth muscle cells and of mouse tracheal rings. So good evidence that Splunk was actually functioning physiologically to regulate both calcium signaling and smooth muscle contraction. So she also did studies in airway smooth muscle. So she knocked down ORI by shRNA and found, so this is calcium, and so in wild-type cells, Splunk will inhibit calcium signaling by about 50%. And once she knocked down ORI, this effect was abolished, which I thought was good evidence that Splunk was affecting ORI-mediated calcium signaling in the airway smooth muscle. Here she did surface biotinylation. So here's the western blot of Splunk in the plasma membrane. And again, you can see that Splunk is actually causing about a 50% reduction in plasma membrane ORI over time. So at this point, we then, as we did with ENaC, we wanted to see which part of Splunk was responsible for this effect. But unlike for the ENaC study, since we had Splunk's crystal structure and had a whole bunch of Splunk mutants, it was actually a lot easier to do. So again, so this is the crystal structure of ORI with the different regions. So there's 6 alpha helices and a beta-pleated sheet area. So interestingly, I didn't mention this, but mouse Splunk actually doesn't have the S18 region. So mouse Splunk doesn't regulate ENaC. And I don't think ENaC is regulated by Splunk in mice like it is in kind of like larger mammals. But for the ORI effect, both mouse and human Splunk equally gave this 50% inhibition of calcium signaling. So we then took the S18 peptide, which inhibits ENaC. And we also took, so delta 44 is a Splunk lacking the first 44 residues, so it doesn't have that S18 region. And so the peptide had no effect on calcium signaling, and Splunk lacking that region could still inhibit it, so which was good evidence that this was nothing to do with the N-terminus S18 region of Splunk or ENaC. We then took Splunk, which lacked the alpha-4 helix, which didn't do anything. But when we deleted this alpha-6 helix, which is actually the C-terminus of Splunk, so in Splunk, the N-terminus and the C-terminus are fairly close together, we actually abolished the calcium signaling. And then we could actually make peptides of this region and found that we could kind of restore calcium signaling. Matt did some really nice docking studies. So the crystal structure of ORI is known, and this is the C-terminus of the alpha-6 region of Splunk. And it basically fits quite nicely into this kind of binding pocket here on ORI. I didn't mention that while ENaC as a whole has very large extracellular loops, there isn't very much of ORI that actually sticks out into the apical membrane. But it looks like there's this potential binding pocket here where Splunk and ORI could interact. So focusing on Splunk-derived kind of alpha-6 peptidomimetics, if you will. So these are some calcium switch studies which we have under review. So this was done in a plate reader actually now. So we switched from using microscopes to plate readers to use calcium, to measure calcium. And we're using Flow4 Direct. So here we're stimulating with thapsic-argin to cause calcium release from the ER. And when we do this in the absence of any extracellular calcium, you get ER calcium release, which is shown here. We then add back extracellular calcium, and the black line is calcium influx through ORI, and this is in HEX cells, I don't think I mentioned. And then if we pre-treat with alpha-6, you pretty much abolish that calcium influx, which is shown here. So again, alpha-6, Splunk-derived alpha-6 peptides inhibit calcium influx. So this is under review, but again, I apologize for not going through all the data, but just in the interest of time. So surprisingly, even though it's a different part of Splunk that's regulating ORI, and I kind of think of it, so the part that sticks out, the N-terminus regulates ENaC, and then the C-terminus, which is back here, will regulate ORI. But we found that basically Splunk will bind to the outside and cause this allosteric. And then again, you get NED42 dependent ubiquitination of ORI, which then sends ORI to the lysosomes for degradation. And again, consistent with that, it takes a couple of hours for Splunk or the alpha-6 peptides to have their inhibitory effects. And if you put it on immediately, you don't see anything. So again, which is consistent with this kind of internalization effect. So to go into a bit more detail in ORI, so I'm going to switch gears now in the kind of latter part of the talk, and rather than talk about airway smooth muscle, talk about immune cells. So as I mentioned, ORI is important in the immune response. And so imagine this is the plasma membrane. So this is a T cell. So when you get the antigen-presenting cell activating the T cell receptor, you get a second messenger cascade, calcium released from the endoplasmic reticulum, and then STIM1 will migrate to the ER plasma membrane junction where it activates ORI. Calcium comes into the cell, you get activation of calcineurin, and then this protein NFAT gets dephosphorylated, moves to the nucleus where it can change gene expression and be pro-inflammatory. And we wondered if using alpha-6 derived peptides to inhibit this, if we could dampen down inflammation in the lung. And so we then went to try and optimize the alpha-6 peptide, and this is new unpublished data. So Florian Sassani in the lab set up a high-throughput screening protocol to do this, and we made a large peptide library derived on alpha-6, and the screening criteria are to optimize for size, then optimize for stability and potency, have a look at solubility, and since any peptides we develop would be inhaled into the lung, make sure they didn't bind to mucins, and then do the basic in vitro and in vivo studies. And this is actually some of the actual data shown as a heat map. So this bluey purple is kind of high calcium, and then as you go through the green, yellow, and red, you get lower calcium. So the vehicle, because there's no agonist, calcium is low. If you add thapsic argon, it goes to this purple region. And alpha-6, as I showed you in the data, gives about a 50% inhibition of global calcium, so which is shown here as this green one. And by optimizing, we were able to get some peptides which looked like they were behaving a bit better. For example, these red peptides looked like they had better calcium inhibition. So we took some of these leads. So the alpha-6 peptide, so towards the start of the talk, I showed that Splunk was highly susceptible to neutrophil elastase, which is a protease that's elevated during inflammation, for example, in cystic fibrosis patients. And consistent with Splunk being sensitive to elastase, alpha-6, so the kind of the scaffold peptide, is also rapidly degraded by elastase. And some of the candidates, so 621 was actually more potent, but was rapidly degraded, so we didn't take it further. Then we had these two peptides, 607 and 604, which were more stable. We did more detailed calcium studies, and it turns out, so compared to alpha-6, so 607, which has a decent stability, was actually more efficacious, whereas 604, which was really stable, you actually dramatically lost potency. So we tried to evaluate some of these peptides further. And just briefly, as I mentioned, that NFAT is important in the inflammatory response. We also looked, for example, with 607 as, for its ability to inhibit NFAT. So NFAT is shown here in green, and when it gets activated, it moves to the nucleus where it starts being pro-inflammatory and changing gene expression. And in the presence, for example, of 607, we could actually block NFAT activation. And this is some of the mean data that's shown here. So you see, you get a significant reduction in NFAT activity. We then evaluated this in mice. And for these studies, we used a pneumonia model. And the reason for choosing the pneumonia model is it's a fairly quick 24 to 48 hour study, so we could screen a lot of peptides. And initially, we're instilling Pseudomonas aeruginosa bacteria into the mouse lungs via tracheal addition. And then in this study, we added the peptide about an hour later, but we've also added the peptide 24 hours later, and we've played around with acute versus chronic dosings. And what's interesting is, so if you look at the monocytes or macrophages in the lung, normally there's predominantly macrophages. And so when you add Pseudomonas, macrophage numbers drop down. Whereas 604, which was the most stable, you didn't really see that much of an effect on macrophage numbers, whereas 607, which was decently stable and more efficacious, you actually saw a kind of return to the naive phenotype. And then most pulmonary clinicians will tell you that when you get lung disease, be it acute respiratory distress syndrome or cystic fibrosis, it's the high levels of neutrophils in the lung which are damaging, and then the high levels of neutrophil elastase which can damage the lung. And consistent with there being a increase in macrophages, only 607 basically reduced neutrophil numbers in the lung. And based on this data, we focused on 607. So we ran a panel of cytokines. So IL-6 is a neutrophil chemoattractant, and 607 really reduced IL-6 levels in the lung lavage fluid, and it also reduced other cytokines as shown here. What was interesting is when Saira counted the bacteria in the lung, excuse me, so after instilling bacteria in the lung, we installed 10 to the 7 bacteria. They actually grew, and there's closer to 10 to the 9. So this is a log scale bacteria. But with 607, you actually see a reduction in lung bacteria in the lung, so it's actually helping clear bacteria out of the lung. And then I think consistent with that, we saw less sepsis. So this is measuring bacteria which go to the spleen, and you get high levels of bacteria in the lung, and I think they basically the lung gets damaged and bacteria can go systemic, which is probably contributes to the death of the mice. And we're also reducing systemic bacteria as well. And what we think is happening is, so Saira and Sabri in my lab took bronchoalveolar lavage from and macrophages from the 6 or 7 treated mice, and it wasn't behaving any differently. So I don't think we're ramping up antimicrobial defenses. But what we did see is we see a highly significant reduction in neutrophil elastase activity in the lung lumen. So too much elastase will start to chew up the lung, and I think we're blocking that. And then total protein in the lung lining is a marker of lung damage. And again, that's going down. I'm not showing the data, but LDH is another marker of lung damage, and LDH was also down. And just looking at the histology, so this is normal lung, so these are the alveolar regions, and you start to see this kind of inflammation, engorgement in the infected mice. And again, 607 is restoring normal lung phenotype. So I think by reducing elastase levels in the lung, we're actually protecting the mice and allowing them to give a macrophage-dominated bacterial clearance. So the other thing we looked at, so this is bacterial growth in vitro. So we Saira plated down bacteria, and then over time, shown in black, they'll increase exponentially. 607 peptide had no effect on bacterial growth, and then as control, we did 2 common antibiotics which blocked bacterial growth. So again, it's not working as a bacteria. So the other thing we did is we demonstrated, so Saira and Joe in the lab took bronchoalveolar lavage and stained for ori, and you can show that with 607, you're damping down ori levels in the lung lumen. Here's the mean data shown here. So we are affecting ori. And also 607 is still working via ori. So again, this is calcium influx. So this is ori1 mediated calcium influx into hex cells. And if you knock down ori by shRNA, that response goes away. So it's targeting ori in vitro. And so global inhibition of ori is immunosuppressive. And so ori knockout mice are actually highly immune compromised. The ori heterozygotes, which have 75% reduction in ori activity, are still immunocompromised, but not quite as bad. So in wild-type mice, PA01 is a Pseudomonas strain. So wild-type mice, you could put in Pseudomonas, and then the peptide rescues the neutrophilia. But in the ori heterozygotes, you see a bigger response compared to wild-type mice, and the peptide was without effect. So I think ori is having this beneficial effect on mouse lungs. Sorry, so peptide is having its beneficial effect on mouse lungs without affecting via ori. I think one of the things that's interesting is, so it's been well known in the literature that global ori knockdown is immunosuppressive, but we're not seeing immunosuppression, so we're seeing beneficial clearance of bacteria. And I'm not showing the data, but this works both for gram-negative bacteria, such as Pseudomonas, but also for gram-positive bacteria, such as Staph aureus. And Henry Danahay had pointed out a couple of years ago that ori in airway epithelia is only expressed basolaterally. And so Joe Rennell in the lab has done some studies with that. So if we put fluorescent 607 apically, which is where it would go if it was inhaled, into the polarized airway epithelia, so the cilia would be here, it doesn't bind at all to the cultures. But if you add it basolaterally, it binds quite nicely, and this is the quantified data. So I think that when the peptide is inhaled, it's binding to the immune cells in the lung lumen, but it's not binding to the epithelia. And I'm not showing it today, but Saira has some nice flow cytometry data showing that it doesn't affect the interstitial immune cells either. So it's really acting locally. And so this is light sheet microscopy, which Joe has taken up recently. And the pink is the fluorescent peptide. And you can see it staying in the lung lumen, is not going out of the lungs. So we're still working on the mechanism of action. I think understanding how 607 can be beneficial is going to be a few years of work to actually do. But basically what we think is happening is when you infect the lungs, you're getting neutrophils coming into the lungs, which are releasing proteases and reactive oxygen species to try and attack the bacteria. But they're also damaging the lungs in the process. I think 607, which don't fully understand how it's doing this, so it may be working on macrophages and neutrophils, but may also be working on, there is actually a few T cells in the lung lumen, maybe working on other immune cells as well. And it's damping down the local immune response, but it's having no effect on the global or systemic immune response. And then you're preventing neutrophil elastase-mediated lung damage and clearing bacteria out of the lung. We've also tried this in some other models, which I'm not showing right now, but I'll be happy to talk about it later if people want. So we see beneficial effects in both gram-negative and gram-positive bacteria, consistent with ori being upstream in the inflammatory response. If we challenge mice with house dust mites and basically give them asthma, we still see a beneficial reduction of eosinophilia and reduced inflammation. And then we've also done studies with chronically infected mice. So those mice, beta-ENaC mice, have chronic mucus plugging and bacterial infections. And again, 607 is beneficial and does fine after multiple dosings. And then we also have a little bit of data for viral infections. And again, we're blocking neutrophilia and seeing benefit. So this is kind of almost my last slide, so I'm close to the end. So my conclusions, I think, so Splunk negatively regulates ENaC and ORI1 by inducing NED4-2-dependent ubiquitination. Splunk is highly susceptible to neutrophil elastase-mediated degradation, and overcoming this may be of therapeutic benefit, and that's something that we're working towards with 607. And so one thing I wonder, since there's 1,000 proteins in the airway surface liquid, and there's also a whole bunch of other peptides, I kind of wonder, are there other Splunk-like ion channel regulators which are present in the lung lining fluid, and then also in other, for example, in the gut as well, which have yet to be discovered. So I want to thank people in the lab. So people in my lab, past and present, who worked on this project, everyone who helped out directly on the Splunk project is underlined. Also people at UNC and around the world. And I want to give a special thanks to Jack Studds, who, like I say, when I was starting out on the kind of more biochemical side of my studies, he pretty much made his whole lab available. It was incredibly helpful in getting me transitioning from a new faculty to getting funding for this. This really is my last slide. So I just want to say, advice to new people starting out, be realistic with your studies. Everyone seems to like their data, but you've got to think about, is it going to be something that the grant funders want to see? I know I got lost in the weeds as a new faculty for a year or two before I found Splunk. But don't give up. It's a lot of work, and reviewers and other people always want more data. Then a quote from Francis Collins, who's director of the NIH and a UNC graduate. It's like, if you're not outside your comfort zone, you won't learn anything. And I think that certainly has been true for the Splunk project, where I've had to move into new areas currently. And actually, whilst the lockdown has sucked, in a way it's been good for us, since we've been able to basically retrain the lab towards immunology to study how Splunk affects the immune system. And there's certainly outside my comfort zone, but there you are. So thanks. I think that's that. And I guess if anybody has questions, they can talk to me during the Meet the Speaker session.
And on behalf of the Physiological Society, I want to say thank you very much for giving the talk today. But also, Alan Hodgkin said that research is more like paddling a small canoe on a mountain river. The river, which is fed by many distant springs, carries you along all right, though often in a peculiar direction. And he also said you have to paddle quite hard to keep afloat, and sooner or later, some of your ideas are upset and are carried downstream like an upturned canoe. So I think what you've shown us today is that you've got some skills in paddling a canoe and responding to the new directions and keeping it upright, or at least that's what it looked like in your photographs at the beginning, I think, where there were some real canoes on the water there. I think what you've also shown is a sort of new contribution to our understanding of airway surface liquid and the components that are in there, and how they regulate ion channels to regulate and their contribution to the pathophysiology of airway disease. So thank you very much. It's been a really interesting afternoon, and I know there'll probably be lots of questions. We can't answer them now, but as Rob said, if you'd like to join the Meet the Speaker session, which is immediately after this session, I'm sure Rob and our speaker from earlier on today will be happy to answer questions on their work, but maybe also on how they got to where they are today. So please join us next. Thanks, Rob.
Thank you.